Namaste. Welcome to Shaf's Biotech for you. Today we'll discuss about bacteriophage derived vectors. Bacteriophages are nothing but the viruses that infect bacteria. Types of bacteriophage derived vectors include M13 vectors, lambdaphage vectors and cosmids. Today we'll discuss about the first type, that is nothing but M13 vectors. M13 is nothing but it is a male specific lysogenous filamentous phage which has circular single stranded DNA approximately 6.4 kb in length. This is the diagrammatic representation of M13 vector, which has a LAGZ gene, lag promoter and operator gene, and M13 genome. Construction of the M13 series. M13 bacteriophage, when added upon by LAGZ gene, resulted in M13MP1 vector, which had no restriction site, but it contained GGA-TTC nucleotide sequence at the start. After single nucleotide mutation at G being replaced by A, it resulted in EcoR1 site. This vector is now called as M13MP2 vector. Because even after the single nucleotide mutation, the LAGZ gene is intact, blue-white screening of recombinants versus non-recombinants is possible. If this MP2 vector is added upon by a polylinker or a short nucleotide, oligonucleotide, which has a series of restriction sites inserted into EcoR1 site, it resulted in M13MP7 vector. Even in this vector, LAGZ gene is intact. This happens because the mutation or the insertion of the polylinker is done at the start of the LAGZ gene and not in between. Hence, blue-white screening of recombinants versus non-recombinants is still possible. This blue-white screening is being explained clearly in the previous class. So, M13 vectors can now act as proper vectors with the foreign DNA being inserted at EcoR1 site or at the series of restriction sites. Coming to the advantages. The genome size is less than 10 kb, hence it is easy to handle. They have a simpler infection cycle where the introduction of the DNA can be done through the pili during the conjugation. Then it also contains polylinker and alpha peptides fragments, so easy blue-white screening, which is single-step screening, is possible. Coming to the disadvantages. The gene of interest more than 2 kb cannot be cloned. It results in low yield of DNA and they produce many toxins at high concentrations. With this, I wind up today's session regarding M13 vectors. Please like, share and subscribe to my channel.